Hi, everyone. It's an absolute pleasure to be kicking off the conference this morning. I'm really looking forward to the next couple of days of talks. So I'm going to be talking about some of my PhD work that seeks to consolidate archaeological and climatic data within the context of the Eastern African Middle Stone Age. So this work is actually being published in Scientific Reports today at 10 a.m. GMT. So I'm super excited about that. And I'll be posting links to the paper on my Twitter, which I've put here if anybody is interested. So fossil, archaeological and environmental records, together with the geographic placement of the region, suggest that Eastern Africa played a prominent role in the evolution and dispersal of modern humans. The Middle Stone Age is the phase most commonly associated with the emergence of our species across Africa, and the Middle Stone Age record of Eastern Africa is particularly rich, largely due to the extensive research history and the preservation conditions of the region. Archaeological studies of the Eastern African Middle Stone Age have long proposed a link between the distribution of sites, behavioural diversity, and environmental conditions. However, many of these studies have either used modern biogeographic zones to represent Pleistocene environments, or they rely on simulated data sets representing arid humid extremes, both of which lack chronological precision and assume uniformity across successive glacial interglacial cycles. Proxy records have improved in chronological resolution over the past decade, yet the climatic patterns and the lacustrine records used to generate them are diverse and are rooted within a small number of lake basins, often without stratigraphically associated archaeological assemblages. What's more, the lake cores yield data that act as relative wet and dry proxies, and translating these into tangible climatic or ecological parameters to represent exactly how wet or how dry is challenging. So overall, whilst the region yields relatively rich archaeological and climatic records, there are substantial disconnects between them, which makes understanding the influence of environments on early modern human behaviour very difficult. So our research provides an integration of climatic and archaeology of the Eastern African Middle Stone Age within a spatiotemporally explicit framework. We use new high-resolution climate simulations of mean annual temperature, total annual precipitation, and biome reconstructions, and we extracted the values from the 1,000-year time slice representative of the middle of each assemblage's date range. We extracted the values within a 50-kilometre radius around the coordinates of each site, representing the population's home range. We had 84 assemblages in total and data regarding the presence or absence of 16 tool types and 8 raw materials, as well as other site characteristics like altitude. The archaeological data has been previously published by my co-authors Matt Grove and James Blinkhorn in Archaeological and Anthropological Sciences. So some of the sites have multiple assemblages, and so when these assemblages have been dated to different time periods, we were able to look at how the environments vary both within and between sites across space and time. So from integrating the assemblages within the climatic model, we found that occupations tend to occur in subhumid climates and landscapes dominated by tropical xerophytic shrubland. We performed a cluster analysis to help highlight some of these patterns. The optimal number of clusters was determined to be 10 by an automatic clustering algorithm. And as you can see here, most of the assemblages are dominated by tropical xerophytic shrubland environments. However, interestingly, there are 2 cold clusters where we don't see this biome at all. Cluster 9 especially, which is the coldest group of assemblages, occupy very diverse landscapes, as these are all at high altitude where the complex topography allows diverse biomes to occur closer together than usual. So looking at diachronic and spatial patterns, we found that during MIS 7, there was a dramatic increase in diversity of the environments occupied, with emphasis on forest woodland ecotones and tropical xerophytic shrubland. MIS 5 was marked by another phase of expansion, though with limited presence in woodland habitats and typically warmer, drier, and lower altitude occupations. Alternatively, in MIS3, there was a push into higher precipitation woodland habitats with a limited change in temperature and altitude. However, we then asked, do these environmental changes simply reflect shifts in the underlying ecology, or do they reflect actual changes in habitat preferences? So using a chi-square test, we found that the relative proportions of each biome available across the landscape did not change significantly between marine isotope stages, suggesting that any changes in the biomes occupied does actually reflect a shift in preference. So we then looked at the overall proportions of biomes available throughout the Middle to Late Pleistocene, and we compared these with the proportions of biomes found at Middle Stone Age occupations. The proportions of sites with access to tropical xerophytic shrubland is slightly higher than that found across the landscape, suggesting that Eastern African Middle Stone Age adaptive strategies were centered around engaging with subsistence resources associated with this biome. But most importantly, we found that the proportion of sites with access to woodland biomes is much higher than is generally found across the landscape. We also found that all of the sites without access to tropical xerophytic shrublands were ecotonal, so they occupied the boundaries between multiple biome types. And engagement with new biomes tended to occur on the periphery of the home range. So together, this suggests that when expanding beyond the widespread and widely occupied shrubland settings, populations preferred the rarer areas with access to a diverse range of resources that would have allowed them to sustain themselves. So let's quickly look at a case study. So the youngest Middle Stone Age assemblage at Pangayi Saidi is placed at a unique tropical ecotone. So Middle Stone Age occupation at Pangayi Saidi at the end of MIS5 is rapidly followed by cultural change, with the appearance of the later Stone Age here occurring earlier here than anywhere else in the region. New constellations of tools within MIS5 toolkits suggest corresponding changes in behaviour previously unseen in the Middle Stone Age, which perhaps may have been key for these expansions beyond tropical xerophytic shrubland. Together, this suggests that certain Middle Stone Age toolkits may have led to adaptation, as opposed to adaptations being a prerequisite for expansion, with transient ecotonal habitats requiring the adoption of a range of survival strategies and potentially mediating interactions between different groups across different environmental settings. We also produced phased habitability models to map the extent of the climates inhabited by Eastern African Middle Stone Age assemblages. To do this, we grouped the assemblages into the more abundant interglacial marine isotope stages, and the climatic range produced by each phase subset was projected throughout every 1,000-year time interval for that marine isotope stage. And then we calculated the percentage of habitable cells, so the cells that remain within that climatic range, we calculated to identify areas that were persistently habitable, as well as the geographic range and temporal scope of impersistent habitable landscapes. So here we're looking at habitability models for precipitation in blue, temperature in red, and precipitation and temperature combined in green. And the colour scale represents the percentage of cells that remains habitable within that time period. So these models fundamentally show that interglacial marine MSA occupations, especially in marine isotope stage 5, may have been much more spatially diverse than presently known. Our models suggest that expansions during earlier phases tended to be constrained by the available precipitation across the landscape, whereas in MIS3, temperature may have been more limiting over where MSA populations were living. MIS3 includes very hot periods, so this suggests that occupations tended to be limited to the cooler, high altitude regions, perhaps to mitigate these increases in temperature. So as you can see, the region towards the east of Lake Victoria remains habitable throughout all of these models, and this has important implications for it as a refugia during periods of climatic deterioration. So this region is marked by a fluctuating ecotone between tropical xerophytic shrubland and various forested and woodland biomes. Lake Victoria today experiences very similar environments where it forms an important boundary for large mammal and human populations in the Central African forests and the savanna of eastern areas. The availability of fresh water provided by surrounding rivers and springs and the complex topography of the Rift Valley may also have helped buffer against the strongest effects of climatic change in the area. Occupying refugia likely required minimal cultural adaptation to environmental change, which could explain why assemblages from within the Lake Victoria Basin show distinct differences from the general Eastern African Middle Stone Age. Also, Lake Victoria is the source of the White Nile, and therefore its refugial position could also have interesting implications for dispersals from the region out of the continent. Together, this highlights the potential role of refugia in structuring Middle Stone Age human cultural and biological diversity, laying the critical foundations for later human evolution. Finally, to assess whether temperature and precipitation have an influence over the diversity seen in Eastern African Middle Stone Age toolkits, we performed some matrix correlations. These were done using a number of other variables which have been analysed in the original paper. However, here we have updated the climatic variables with the output from our analysis to represent the environments of each assemblage. We found that precipitation, but not temperature, has a statistically significant relationship with toolkit composition and almost statistically significant relationship with raw material use. Precipitation is widely known to be a key parameter affecting adaptive strategies due to its strong effects on the suitability of the environment for habitation. So quickly in conclusion, because I know I've run over, our application of high-resolution paleoenvironmental reconstructions has enabled a previously impossible characterization of the ecosystems inhabited by early human populations at this scale, with existing site-based and paleoenvironmental reconstructions very useful, but difficult to integrate and translate into a comprehensive framework for the region. Whilst the application of high-resolution climatic simulations to archaeological data is inherently limited by the uncertainty surrounding radiometric dates, and future work should focus on developing methods that address the effects of dating error on conclusions derived from climatic models, our results are important for demonstrating that environmental conditions inhabited throughout the Eastern African Middle Stone Age, with levels of diversity far beyond those typically assumed by classic habitat-specific hypotheses for human evolution. So I'll end it there. Thank you very much for listening, and yeah, I'd be happy to take any questions.
Characteristic in the Cantabrian region, and especially in the eastern area of Asturias. The current state of the art on the exploitation of mollusks, crustaceans, and echinoderms reflects the system of a certain variability in the degree of exploitation intensity during the course of Mesolithic period in the region, so that human population probably adapted their gathering strategies to the specific needs of each moment throughout the 4,000 years that the Mesolithic lasted in the region, 10,800 to 6,700 Cal BP. Based on this hypothesis, the present study aims at analyzing the shell assemblages from the site of el Toral III, Llanes, Asturias, in order to determine species representation, size variations, and collection areas as a first step in the evaluation of intensification processes through time. Toral III is located in northern Iberia, very close to the town of Llanes in Asturias, and at a distance of around 1 kilometer from the current coastline, as you can see in the slide. In 2009, extensive salvage excavation at El Toral III cave brought to light different stratigraphic units, which allow us a better understanding of the formation and post-depositional processes at the site. In the slide, you can see the site when it was found and after retiring sterile and disturbed sediments before the excavation. Despite the heavy erosion that affected some parts of the site, a well-preserved stratigraphic sequence was found. In Area A, units 10 and 13 were identified as in situ. In Area B, units 17, 19, 20, 21, and 22 were also in situ and showed relatively good preservation. Shell-midden stratigraphic units of Mesolithic age produce a variety of materials, including mollusks, mammal, fish and bird bones, lithic, charcoals, seeds, and human remains. Radiocarbon dates and material culture showed the use of a site for long periods of Mesolithic and also as a burial place in later time, Chalcolithic and Bronze Age. Mesolithic units from Area A were dated to 10,000 to 9,500 Cal BP, Ipset Unit 13A, which was dating to the interval 7,900 to 7,700 Cal BP. Units from Area B were dated from 7,900 to 7,280 Cal BP. The results on species representation show the presence of marine bivalves and gastropods, as well as echinoderms and crustaceans. Although there is a certain variability in the species representation, only 4 species account more than 80% of each cell assemblage. These species are the limpets Patella vulgata, Patella depressa, and Patella lysiponensis, and the top shells Phorcus lineatus. When we compare the abundance of each species, we can see 3 different patterns. First, Unit 10 is a small lens at the bottom of the sequence composed exclusively of limpets Patella vulgata, Patella depressa, and Patella lysiponensis. This is probably related to a very short and sporadic visit to the cave. Then, the 3 subunits identified in Unit 13 present a more similar pattern, with larger amounts of top shells followed by limpets Patella depressa and Patella vulgata. And finally, in the younger units located in Area B, assemblages are clearly dominated by the limpet Patella depressa and to a lesser extent Patella vulgata and Patella lysiponensis. In these units, we can observe a dramatic decrease in the amount of top shells Phorcus lineatus compared to the units from Area A. Therefore, there is a significant change in the most collected species through time. These results are in agreement with previous data from the nearby site of El Mazo, where a similar pattern was found. The explanation from this change is probably related with the viability of the different species due to changes in the climatic and environmental conditions of the area through time. When we observe the size distribution for each species, 2 patterns emerge. First, units 10 and 13C from Area A present larger sizes in Patella depressa than shells from units 17 and 22 Area B, which present very similar sizes between them. A very similar pattern is visible in the size of Patella vulgata. However, the pattern changes when it comes to Phorcus lineatus, that show a slight increase in the sizes from older to younger units. The limpet size decrease might be related to intensification in the collection of these species through time. However, the pattern showed by the top shells is more difficult to explain in terms of intensification. This pattern contrasts with the results from other sites like El Mazo, where a similar trend in shell size through time has been recorded. Therefore, further research must be conducted on that question in order to explain the different pattern between the 2 sites. Studies on zonation and exposure have been applied only to Patella vulgata using the equations established by Greathead and Bally. The results show that around 80% of the limpets were collected in the lower shore. Meanwhile, in the case of exposure, the equation revealed that collection took place almost exclusively in spot shores, 90% to 98%. Collection of limpets in lower and spot shores has been previously identified with intensification in shellfish gathering. In the slide, you can see the exploitation areas of coastal resources nearby Toral III cave. The beaches of La Vallota and Andrin are next to each other and around 1 kilometer from the archaeological site. Our research concludes that the systematic use of mollusks and the significant amount of shells recovered on each shell midden unit points out to an intensive collection of intertidal resources throughout the entire stratigraphic sequence. In terms of species representation, there are differences in the shellfish gathering patterns through time, probably related to the viability of the different species due to climatic and environmental factors. The decrease in the size of the limpets between the oldest and younger units is probably related to intensification in shellfish gathering through time. However, collection in lower and spot areas has been recorded throughout the sequence, which indicates that some kind of intensification was acting since the initial occupations at the sites. And finally, thanks all for your time and attention.
Center in the same institute, which is the Indian Institute of Technology, Gandhinagar, in western part of India. So what's in a bone? The bone is enclosed in a matrix, which is 30 to 40% of organic material, which is basically collagen, and the majority of the bone matrix is the mineral dialite. Studies of bones are important as we can get a host of information regarding the paleoclimate, paleopathology, and even genome sequencing. However, diagenesis can hinder the estimation of these parameters, as we all know. What is diagenesis? Very quickly, it is essentially the physical and chemical variation of the bones after burial. These can be classified on the basis of morphological, chemical, and organic variations. Morphological ones include cracking and color change. Majorly, chemical variations include exchange of elements with the surrounding ground water or soil. And the diagenesis is also quite dependent on the environmental conditions under which the bones are buried. These include the acidity or alkalinity, the pH of the soil, temperature, organic content of the soil, among many other factors. So in this study, we have used the following instruments to characterize diagenesis parameters. The stereo microscope looks into the surface shell alterations, while the X-ray diffraction and the X-ray fluorescence were used to characterize the chemical diagenesis. Our study area is Dholavira, which is one of the largest Bronze Age period, or in India, we call it the Harappan civilization or the Indus civilization site. And it was recently declared as a UNESCO World Heritage Site, and it's present on an island called Khadir Island, which is basically salt pans with a very unique environmental set of conditions. So this this area is called the Run of Kutch, and it's in the western part of India. The environment is extremely arid with annual precipitation of 262 millimeters per annum of rain. So here is the view of the island, and the site is around 5,000 years old, and it is very well known for its exquisite town planning, because the site is very clearly divided into sectors like the castle area surrounded by the bailey, the middle town, and the lower town, where the different divisions of societies resided. And it was occupied for a very long time period through the earliest phases of the civilizations to its maturity and the decline or the deurbanization phase. So these are some of the ancient and modern bone samples that were collected for the study. B1, B2, and B3 are the ancient samples, while M1 or M is the modern sample, which was collected from the surface of the site in one of our recent fieldwork or explorations. So B1 is the distal epiphysis of the metapodial of a horse, and it comes from the castle area. B2 is a mandible fragment of domestic cattle from the lower town area, and B3 is a mandible fragment again of sheep or goat, which also comes from the castle area of the site. The modern bone is a left side scapula of domestic cattle, and it was found lying just outside the lower town. And we assume that this modern animal could have died anywhere between 0 to 5 years in the last 5 years, whereas the ancient bones were definitely 4,000 to 5,000 years old. The initial studies from the stereo microscope indicate a number of manganese stains, which are depicted as black spots here. The ancient samples also have indications of calcite infillings, which is not present in the modern samples. So this is what we majorly observed under the stereo microscope. The XRF studies show characteristic exchange of elements for both the modern and ancient samples. The ancient samples are observed to be more altered as compared to the modern ones, which is indicated by the anomalous concentration of iron, strontium, arsenic here, and also chlorine. This has been correlated with the sediment sample, which is collected from the same area. We have these 3 rocks, which were also analyzed through XRF. And these are, they also show the same kind of elements. The XRD results depict substitution of secondary elements such as chlorine and samarium. Further studies are required to derive the origin of these elements, however. Moreover, bone, they have an amorphous structure, and on diagenesis, they get crystallized. The greater the crystalline percentage or crystallinity percentage, the greater is the amount of diagenesis of the bone. So, in conclusion, sorry for the animation. So in conclusion, the stereo images provided evidence for surface etchings and discolorations for both the ancient and modern samples. We also saw some amount of weathering according to the Brenn's stages, 0 to 2. Furthermore, certain samples also do contain mineral infillings, as we saw. XRF studies indicate incorporation of a number of elements such as arsenic, barium, bromine, strontium, cobalt in the ancient samples, which could be attributed to the exchange from the sediment and groundwater samples, which form the matrix for these ancient bones. Hypotheses can be tested if these can be correlated with each other. There's also a loss of hydroxyapatite and substitution of secondary elements such as chlorine and other trace elements. Ancient samples definitely exhibit relatively greater crystallinity. Investigations suggest some order of diagenesis for both the ancient and the modern samples, though the ancient samples have undergone greater variation in chemical composition. We definitely propose a use of caution for the strontium analysis, because we have elevated values for strontium, which can create problems for analysis further on, especially for isotopes. And we don't really know the source of this elevated strontium values. Preliminary mechanism indicates chemical incorporation from the sediments in groundwater to the bone. So in future, we definitely would like to continue with this analysis, not just with these 3 bones, but also with more number of bones from different parts of the sites. And we would like to concentrate on not just on the FTIR analysis, which will give us a better understanding of the collagen presence or absence, but we would also like to put these bones through ICP-MS for rare earth element analysis, and more isotopic and trace element analysis for the source identification of these elements. So these are some of the references that we have referred to during the work. And thank you.